So we start to talk today about the brain, finally. Okay. And first, we will discuss the development of the brain. Interestingly enough, brain develops from so-called neural tube. I deliberately, I removed the picture of the neural tube because honestly, it looks like a tube. Really. So it closes on the proximal and distal ends during the initial embryonic development. And then the first stage of development is formation of 3 brain vesicles. Okay? The prosencephalon, the mesencephalon, and rhombencephalon. Okay? Which is forebrain, midbrain, and hindbrain. Each of these portions of the initial neural tube develops further, and the secondary brain vesicles, embryonic brain vesicles, are telencephalon, or Mickey Mouse head. Okay, telencephalon develops into the brain, the cerebrum, the cortex. Okay, that makes sense? That's the thinking part. Right? And also it forms ventricles, lateral ventricles, and we're going to discuss what these ventricles are. Diencephalon keeps the name, and it develops in diencephalon, which consists of thalamus, hypothalamus, and epithalamus. And also there's third ventricle located in between. And mesencephalon develops in the midbrain. And in the center of the midbrain is cerebral aqueduct. Metencephalon develops in the pons and cerebellum. And myelencephalon develops in the medulla oblongata. These two also give rise to the fourth ventricle. The posterior part of the neural tube develops into the spinal cord, and in the center of the spinal cord is the empty structure called central canal. As we go forward, you will see the difference between ventricles and the brain tissue. Now, when the brain grows, it's not straight. As the neural tube develops, it's not just a straight tube. It flexes. You can see it forms cervical flexure and midbrain flexure. Okay, and as it grows, the cortex, the hemispheres of the brain, they grow backwards and lateral. Okay, so you can see it here. They grow in the way that they wrap around the midbrain, which is shown here in purple. And as they mature, about week 26th of pregnancy, they're pretty much, similar to what we see in adult. Brain, here's the thing. So brain, first of all, it wraps around the midbrain, the diencephalon and the midbrain. But the thing is, the cranium, your skull, come on, Melvin. The cranium is smaller than the brain. So brain, in order to feed the cranium, has to fold. Does that make sense? That's where those folds are coming from. Now, folds are not just because it doesn't fit there. Folds increase what? Surface area. And surface area means more neurons in that surface. And it turns out, cortex, the part of the brain that's responsible for your voluntary actions, is the cortex. Does that make sense? Yes. Say again. So what I'm saying is that it is shown here as a straight, but this is more what it looks like in the upper right corner. what I'm saying is that it's not like lying like this, it's more like bent over. Does that make sense? But it still consists of all those vesicles. Imagine that you're looking at it from the top. So you don't see those. So look at it from the top, you don't see all those folds. Does that make sense? Now, all right. There it is. Ventricles. So, oh crap, I forgot mine. Anyway, look at this horny things. Can you see them? The gray ones, the horny ones. Yep, these ones. Those are brain ventricles. Now you have to appreciate that ventricles are empty spaces inside of the brain. Does that make sense what I'm saying? So they're not like gray mass. They are chambers inside of the brain. So here in this picture, they're shown in blue. So imagine those are empty spaces that are filled with blue liquid. And no, liquid isn't blue, just for the purpose of illustration. Now, if you would look at your model, you can see 4 ventricles. Okay? You can see 2 lateral. Can someone identify lateral ventricles for me and show them to me? Lateral ventricles. That's one, and that's another. Okay? Can you show it to everybody? Thank you, Jacques. So that's one lateral ventricle, and that's another lateral ventricle. Okay? Now, between the ventricles is interventricular foramen. That's the mouthful, and you kind of can see it in the picture right here. Foramen is a little sort of a channel that connects these 2 lateral ventricles. Generally, in anatomical terminology, foramen, what does that mean? Did anyone take anatomical med term? Foramen? Foramen, no? No? No foramens in med term? It's a hole. Generally, foramen means hole. So, in this case, foramen is the hole between 2 chambers, which allows the flow of cerebrospinal fluid. Okay? A little bit down from lateral ventricles is the 3rd ventricle. Can you identify the 3rd ventricle on your models? Can you see the 3rd? Yeah, it's the whole, it's like big, looks like a rooster head, like a bird head. Okay? Can you see that? With a hole in the center. That's the 3rd ventricle. Okay? Now, 3rd ventricle is connected by the cerebral aqueduct, or I think it's called aqueduct of Sylvius, some dude, to the 4th ventricle. Right here. Can you see the cerebral aqueduct and the 4th ventricle? 4th ventricle is that tail you can see on the very bottom. There's the 4th ventricle. And 3rd and 4th are connected by the cerebral aqueduct. Now, 4th ventricle is connected further to the central canal of the spinal cord, but we don't have it on the model. Does that make sense? So far, anatomically.
That's essentially how the cerebral spinal fluid gets into it because it's connected to the spinal cord.
Getting there. We're getting there. Okay? So far, anatomically, do you get the chambers? So you have lateral ventricles, 3rd between them, 4th in the bottom, cerebral aqueduct connects the 3rd and the 4th. 3rd and lateral are connected by foramina, interventricular foramina, and the 4th is connected to the spinal cord. Okay? CSF, cerebrospinal fluid. It's produced by the structures called choroid plexuses. Okay? On these models, you can see the location of choroid plexuses. They're labeled by pink color. You can see that there are choroid plexuses, a lot of them in the 3rd ventricle, in the 4th ventricle, and they're not shown on the model in the lateral ventricles as well. Can you see them? Can you see the pink labelings? Okay. So before we move on, I want to ask you a question. Which bodily fluids do you know? Do you know? Saliva? Yeah, because, well, it's not so much fluid, but, well, all right. Okay, a little bit of a fluid in the lungs, yes. Not much. Serous fluid, good. What else? Urine. Spinal fluid, blood. Okay. Now, there's one, like, absolutely major fluid, right? It's a blood. Where do other fluids come from? Like urine or CSF? Huh? Exactly. They come from blood. If you will look at every process for the fluid formation, turns out that essentially blood is being filtered. Every time, like when saliva is formed, all the cellular elements of the blood are filtered out. Plasma goes through the serous cells of the salivary glands. Filtered, slightly altered chemically, you've got your saliva. Urine, it's a product of blood filtration, okay? CSF is the same. If you would look at the structure of choroid plexus, you will see that there are capillaries right here, okay? Can you see capillaries? And capillaries, the walls aligned with ependymal cells. Ependymal cells, and we mentioned them before as the special cells in the CNS, ependymal cells serve as filtering cells, okay? So they don't let cells of the blood to get into the ventricles. They slightly modify plasma and produce cerebrospinal fluid. Does that make sense? So cerebrospinal fluid is produced in the ventricles. So it fills the whole thing. It fills lateral ventricles. It fills third ventricle. Via cerebral aqueduct travels to the fourth ventricle. And in the fourth ventricle, what happens is pretty exciting. In the fourth ventricle has median and lateral apertures. Okay? From the fourth ventricle, cerebrospinal fluid has three exits. One exit is to, central canal of the spinal cord. Does that make sense? Another is through median and lateral apertures into the so-called subarachnoid space. A very narrow space that surrounds your brain and spinal cord. So if you try, and this picture kind of shows you here. Look at this. You have a brain that is immersed in the fluid. So this blue liquid is the fluid. Does that make sense to you? Brain is immersed in the fluid. Okay? As well as spinal cord. So when you get the spinal tap, well, I hope nobody did, but if somebody gets the spinal tap, or lumbar puncture, the proper term, the needle is inserted at the level of about third lumbar vertebrae into the subarachnoid space. And you see that this whole empty space, that whole space that CSF fills up, it is continuous. So the same cerebrospinal fluid that surrounds your brain circulates and at some point reaches the spinal cord. So it all circulates. Does that make sense? Do I understand that idea? Do you understand that concept? You see? Yes, bacteria in the meninges, bacteria in the cerebrospinal fluid White blood cells in the CSF, low glucose in CSF is the sign of bacterial infection, usually meningitis. Now, what's the function of CSF? Well, kind of written, cushions the brain. Not only this, if, like, did you try to, go ahead. Say again? Nourish, not that much. It's mostly blood. It takes some of the crap away, but not that much. It does, because it comes from the blood, so it does have some organic matter. So it does have some sugars, some respiratory gases, but not a lot. A little bit of nourishing. Main function is cushioning and floating the brain. Did you ever try, when you were kids and you played with other kids in the water? If you try to lift someone in the water, it's really easy because of flotation, right? Cerebrospinal fluid decreases the weight of the brain by 97%. Okay? Now, to give you better appreciation of how it looks like, the layer of CSF that surrounds the brain is very thin. If you want to have a perception how thin it is, take a penny and look at the side. That's it. But that's enough for the brain to be pretty well cushioned in the solid cranium. So every time you do jumps or any types of accelerations or head movements, your brain doesn't quite literally hit your cranial box. Does that make sense? Every day, choroid plexuses produce about half a liter, a pint of CSF. However, the total volume of cerebrospinal fluid in circulation at a given moment, normally, is 150 mils, a little bit less than a cup. Which means it constantly drains out. Does that make sense? Cerebrospinal fluid drains out through the structures called arachnoid villi into the veins that go through your brain, so-called sinuses. Before we move on, do you understand the idea? Choroid plexuses produce CSF, and CSF drains through the arachnoid villi into the blood. CSF is derived from the blood and it is returned into the blood. You wanted to ask about hydrocephalus? No? Okay. If it comes up, so what happens if arachnoid villi are blocked? Drainage of the fluid is blocked. This is called hydrocephalus. Accumulation of cerebrospinal fluid in the brain. I don't know, now it's not really a frequent thing because it can be quickly diagnosed and treated. But you may have seen children with abnormally enlarged skull, maybe adults with abnormally enlarged skull. Usually it's elongated and just abnormally large. Maybe a sign of the hydrocephalus. Accumulation of the fluid happens because there's no drainage, okay? It can happen due to various reasons. Most often it's either trauma or brain tumor. Now in kids, CSF, or newborns or very, very, infants, accumulation of CSF in the brain usually, comes, and goes easier than in adults. Any idea why? The skull is informed. The skull is soft. So, so-called fontanelles, little joints between the bones of the skull, can stretch. that stretching increases the size of the head, but it sort of reduces the pressure that excessive cerebrospinal fluid can put on the brain. In adults, it's really a life-threatening condition. It's a serious condition. How do you think it is treated? Well, in acute treatment, you can do that. Yes, you can drill a hole in the head and drain the CSF, but the problem is that often, if it's a trauma or some other mechanical damage to arachnoid villi, you have that problem forever, right? And I bet it's kind of inconvenient to walk around with a hole in the head. Something drips out of it. They do drain it.
So they replace part of the skull with a plate.
Not really. They actually drain it. Abdomen, yeah. They drain it into the abdomen. Like really, like abdominal, peritoneal cavity. So the person has pretty much the tube installed permanently. Does that make sense? Because you cannot drain it back here because it's all connected. You can't just move from one side to another. It's going to be a disaster. So instead, this fluid is constantly drained into the abdomen. That makes sense? Anatomy. I'm going to test you on that, on anatomy. I will give you the list of anatomical parts. So for ventricles, absolutely. Interventricular foramina, for sure. Cerebral aqueduct, for sure. And the horns, so you have inferior, posterior, and anterior horns of the lateral ventricles. That's pretty much it in terms of the anatomical terms that you have to know. Am I clear? And, you know, what is the role of the choroid plexuses? What's the role for arachnoid villi? What is the source of the fluid? It's blood. Okay, does that make sense? So, what Jacques mentioned, meninges. Okay, meninges are layers of connective tissue that surround the brain. There are 3 meninges. Okay, dura mater, and I go from superficial to deep. Dura mater, arachnoid mater, and pia mater. So in this image right here, you can see the skin. It covers the scalp. Well, skin off the scalp. That's what people lose when they're being scalped. Periosteum, the connective tissue membrane that covers the bone of the skull, and then first layer is dura mater. Okay, it's the thickest layer thickest of all the meninges. Okay, consists of connective tissue, very strong layer. Okay, and dura mater in itself has 2 layers. One layer, the layer that is attached to the bone, is called, I'm going to color it in blue, this layer is called periosteal. Periosteal next to the bone. Does that make sense? Periosteal layer of dura mater. And then there is a meningeal layer, which is deeper than periosteal. When periosteal layer and meningeal layer separate from each other, at the site of separation, they form special blood vessels that are called dural venous sinuses. These venous sinuses, they drain the blood away from the brain. That makes sense? Now, why are they called sinuses and not just veins? Look at the shape. They're triangular. Cross section, it's triangle. Vein is usually kind of sort of more or less circle, oval circle. These guys are triangular. Does that make sense? Okay. Dura mater also forms dural septa. Those of you who watched Game of Thrones may have known that there's like septa, septum, all those words. Septa means a wall, a separation. So dural septa, it's the structure that partitions the skull. Like instead of having one gigantic room in your house, you have several smaller ones. Does that make sense? Like internal walls. Okay, dural septa holds the brain in place. So all the structures hold the brain in place. Now, anatomically, be able to figure out dura mater versus, other, and 2 layers of dura mater. Know the function. Okay. Peristitial layer, meningeal layer, what happens when they separate, what is a dural septum, and stuff like that. Okay. Arachnoid matter. You can see on this picture, it's a very thin layer of connective tissue with the projections that connect arachnoid matter to the pia matter. Arachnoid means related, something related to spiders. So it's like web-like layer. You can see that there is a space between the arachnoid matter and the pia matter. That space right here is called subarachnoid. In the subarachnoid space, there is cerebrospinal fluid. It circulates through it. And there's a bunch of blood vessels. Vessels that actually feed the brain. Does that make sense? And then there is pia matter. Okay, pia matter is very, very tiny, very thin, very gentle connective tissue that wraps the brain. This layer, this beige layer. Do you see the pia matter? Let me show it to you again. That's pia matter. Not the brown, but beige stuff that wraps the brain around. That makes sense? Now, if you would look closer, you would notice that arachnoid villi, the structures through which CSF drains into the blood, arachnoid villi are formed by arachnoid matter. Now, since the largest blood vessels are in the subarachnoid space, what's going to happen if one of those blood vessels bursts? Blood, yes. Where? In which space? It's in the subarachnoid space. It bursts. It's still in subarachnoid space. Does that make sense? Ever heard the term subarachnoid hemorrhage? Well, if you haven't, that happens. In case of head trauma, subarachnoid hemorrhage is a real life-threatening condition. The main problem, you cannot diagnose it just by the appearance of a patient. So what can happen, you can have a person who kind of hits his head or her head and, dizzy, and you think, oh, it's probably concussion. He or she should be fine. They will sit on the sideline. Like it's a soccer game, okay? A couple of hours, this person's dead. Because the bleeding, it's not about the blood loss. It's about the accumulation of the blood in a very tight space and the pressure that's exerted on the brain. Does that make sense? Go ahead.
So it's just like any other aneurysm in the brain, but it's just like it's just the location of it.
Say it one more time.
So it's just like if anybody has an aneurysm that
Right.
occurs in the brain,
Yep.
and the pressure, like the pressure is what does the damage. It's just the location of it is what makes that different.
Well, usually subarachnoid hemorrhage can arise, well, not usually, but subarachnoid hemorrhage may arise not from the bursting aneurysm, but just from the rupture of otherwise healthy vessel. Really hard hit. Does that make sense? Because what Jacques refers to is the problem called aneurysm, when the wall of the artery becomes extremely thin and the spike in the blood pressure can cause artery to rupture. And blood starts to spill into the brain, and in case of artery, because the blood flow is so intense, it can actually spill into the brain parenchyma, in the tissue of the brain, cause inflammation and death of the brain tissue. Okay? When you refer to aneurysm, you refer pretty much exclusively to spike in the blood pressure. Subarachnoid hemorrhage may arise from the brain trauma. Inflammation of the meninges is called meningitis. Get your vaccines. Meningococcal, pneumococcal, and Hib. Those are three major causes of bacterial meningitis. If you want to know, infection with meningococcal meningitis, Neisseria meningitidis, can kill a person in 24 hours. If it makes it to the brain, the person can be toast in 24 hours. Really hard hit into the hand. You try to play football without a helmet. Okay, like that's why one of the reasons why there is traumatic brain injury in professional football players. Even if they don't have a massive subarachnoid bleeding, they may have tiny vessels bursting that will cause inflammation, that will cause the brain damage, the death of the brain tissue, and all the consequences down the road. I didn't watch the movie, but it's kind of well, I mean, the movie is kind of what are you making a movie about? We all know that people get hit in the head. We all know that they develop brain injury. We all know that they, you know, have health consequences related to it. We all know that NFL acts like dicks, not providing health insurance. That's it. I told you in 2 minutes. You want to spend like $20 on popcorn and tickets and watching 1.5 hours of that? Well, Will Smith, I understand, but does it justify $20? Really? So the same happens to the professional boxers. If you read about them, many of them, when they retired, they go totally bonkers. And part of that may be physiological, just because of the brain injury. So, the brain, hemispheres. Hemispheres are like that light beige stuff, okay? That's hemispheres is 83% of your brain by mass. If you would look at these guys, you can see that brain has elevated parts, the ridges. Ridges called gyri. If you like Greek food, gyro and this gyri, they come from the same root. Same origin, really. Okay? Gyri are separated. Gyri is the plural. Single is going to be gyrus. Okay? So gyri are separated by the very narrow and deep crevices that are called sulci. And singular is going to be sulcus. Okay? Now, if you will, handle the brain and just roll it around, you will see that hemispheres are separated by the very deep line, very deep crevice that is called median longitudinal fissure, or as one of my former students named it in one of the quizzes, the butt crack of the brain. Please don't use it as the answer in the quiz, okay? So median. Now, what's the difference between the sulcus and the fissure? Fissure is deeper. Fissure is the really deep crevice. Does that make sense? Okay? For instance, all those crevices on the surface of the brain are sulci. But this one, between the cerebellum and the cerebrum, is called fissure. It's a transverse cerebral fissure that separates the cerebellum from cerebrum. So, be able to tell apart sulci from gyri, okay? Left from right hemisphere, people, I'm going to ask, and you're going to have a question where you will have to identify whether it's a left or right hemisphere. Am I clear? Okay. Median and transverse fissures, median longitudinal and transverse cerebral, and finally lobes. So brain is separated into 5 lobes. This separation, I'm not a neurosurgeon, so I won't be able to show you on every brain sample that you will ask, the central sulcus. It's probably right here, central sulcus, okay? But generally, you can see the difference. Look, since I don't have a model, I'm going to show it on myself. Each hemisphere has a front lobe. On this picture, front lobe is shown in pink. Parietal lobe, which is shown in light blue. Occipital lobe in the back, which is shown in very light blue. And temporal lobe, which is shown in dark blue. Can you tell the difference? Right? Now, here's what can happen on the anatomy exam. You may have a photograph of that model, and there will be an arrow pointing at the brain, and you will have to pick one answer. Which lobe is that? Does that make sense? Now, I am very distinctive with what I'm pointing at. What I mean, imagine for a second that you have an image of the brain right in front of you, and I ask you, which lobe is that? Occipital. Or, which lobe is that? Or, which lobe is that? Parietal. Or that one? Temporal. I will not ask you something here. Guess what? Which one is that? Well, so I will be very, very clear, but you have to know those 4 lobes. I mentioned that there are 5 of them. The 5th lobe isn't seen on the picture, and you can't really see it on the model. The 5th lobe is called insula. Okay? So it's deep, like right there. Right, deep to temporal lobes. Does that make sense? Lobes are separated by the relevant sulci. So for instance, central sulcus separates frontal and parietal, okay? Lateral sulcus separates temporal and parietal. Preoccipital sulcus separates the parietal and occipital. Does that make sense? So please know your lobes, right? Now, before I will let you go, I want to say a few words. We're not talking about this. Just lobes are isolated, not because just some weird fantasy of anatomists. they actually, you will see that lobes have groups of functions that are unique to each lobe. Okay? There are some functions that are in the frontal, some functions that are in the occipital, parietal, and temporal. Are we clear? Okay. Before